So welcome everyone to this month's ReproNim webinar. We're delighted to have Anna Manelis and our own Jarek Helchenko to present. As I foreshadowed in last month, we're doing a little bit of a change from our original technical presentations to try to feature a little more biology and applications in these presentations. So we both get a dose of the way things are being used or projects that are being done, and a dose of how ReproNim technology either can or is or should be, could be thought about being used in these presentations. So without further ado, I will turn the floor over to Anna and Jarek for their presentation. Thank you.
All right, thank you, David, for introduction. So we'll talk today about the two very important questions, depression and obesity. And we'll show how we applied ReproNim technologies to study these two problems. Okay. Unfortunately, almost 2 billion of adults are overweight and 650 million of them are obese. And over 264 million of people worldwide suffer from depression. So both are serious public health problems. Depression and overweight obesity often co-occur, and they share biological pathways. Because studies show that the same brain regions show some level of dysfunction in depression and overweight obesity, we thought that these two could actually interact and worsen the problem or maybe facilitate the problem in some cases. So the agents like dorsolateral prefrontal cortex and IFG and ventral striatum insula and triacingulate cortex, parahippocampal gyrus precuneus were found to be to have aberrant functioning in both disorders. Very frequently, people focus on task processing or on a food stimuli processing when they study overweight and obesity. So, for example, they could show the stimuli of specific foods, or people can even consume some foods during the scan. But from our point of view, food processing is not the only variable that affects our ability to cope with, specific food, food problems. There is also our anticipation of food. So during anticipation, people might imagine food items. They might want those food items, but they're not really exposed to food items. And I bet, like most of us would have this image somewhere like in the middle of the meeting when you're going to have a dinner, kind of like anticipating having that juicy piece of steak in an hour or two. So in the ReproNim framework, there are 5 steps that every study should go through to get to like a final publication stage. It's a study design, it's a data collection, data processing, statistical analysis, and publication. And while like every single study would go through these steps, we want to make it reproducible, and we want to make it kind of like comprehensible when in 10 years you get this data set and you can actually figure out what was done and how it was done and why. So this is the study design. The study was approved, was funded by NIH, approved by IRB, and it had 2 stages. One was the scan session. The other one was session outside the scanner. During the Fmri session, subjects were presented with food and object items. Each of these items would be preceded with some shape. For example, subjects could be instructed that you will see a triangle. Think about a food pyramid. During this triangle presentation, try to prepare yourself to process food stimuli. And then the food stimulus would appear, and subjects would need to make a judgment of if that stimulus is pleasant or unpleasant. And the same type of trial we would have for objects. We would show the subjects a circle, would tell them, okay, like think about objects, like circle is like the letter O for the objects. Prepare yourself mentally to process object items, and they would be shown the object item, and again, they would have to make a pleasant, unpleasant judgment. So subjects would be participating in 2 runs of this task. They would be anticipating, mentally preparing to process either foods or objects, which would go in a random order, and we use 2 different sets with objects and foods, make the judgments. After they finished the scan, they would be shown the same food items as they have just processed during the scan session, and they would be asked, like, how much do you like this item in general, for example, this piece of cake? And they would rate them between 1 and 9, with 1 not at all, like I don't really like this item, and 9, I like it a lot. The second judgment about the same item would be about how much do you want this item right now, right at the time of the test. And again, they would give a rating between 1 and 9. So the data were collected on the Prisma scanner, and we used Fmri sequence naming according to ReproNim convention. So the naming consists of several pieces. Each would inform about type of a sequence, acquisition parameters, or the task name. So the first field, and I'm talking about this because this is something that makes your life easier when you want to then build up BIDS data set, and what would later help you to tease apart different sequences, like in 10 years when you get the same database and try to analyze them again. So the first field informs you about which sequence was used anatomical, field maps, or functional. Another field would inform you about a specific task or specific task name. So for anatomicals, it could be T1 or T2, and for functionals, like this specific task was called encoding, oops, I said faces, but it's actually encoding food. Another field would inform about acquisition parameters. For example, for field maps, it could be spin echo images collected in anterior to posterior direction, or in the posterior to anterior direction. Or let's say functional sequences were acquired with multi-band factor of 8 and consisted of 2 runs. And this is the first session because we actually have 2 sessions. This is a longitudinal study. So like after the data collected within the specific naming convention, it's easy then to convert those files into BIDS data set. For that, we use Singularity Container. And I mean, like you can use it in 1 step, we used it in 2 steps. So like first kind of like get into the Singularity Container, and then we applied HudiConv that used HudiConv heuristics to convert the files with that specified naming into the BIDS data set. So when the data were converted in a BIDS data set, it would look in a kind of like a conventional BIDS data set structure with the subject at the highest level, and then the session number at the second level, and then another field maps or functional folders at the third level. And inside each folder, we would have JSON files that would describe all the acquisition parameters, as well as the Nifty file that was converted using DCM to NIX. So for functionals, there's also events TSV file that describes when specific event was acquired, in which order, and how long the event lasted. After the data were converted in the BIDS data set, again, it was easier to apply such pipelines as MRI QC and OFMRI prep. Again, we use the Singularity Container, and we use that at the participant level both times. What's convenient is that you can describe the amount of resource that you can allot this specific process. This is especially helpful when you need to process a large amount of data simultaneously and you have limited computational resources. Preprocessing using Fmri prep would involve motion correction, slice timing correction, registration, ICA using Roma, and so on. So the output file would be created in a bits format, and all these little suffixes would inform about what steps were done, what kind of registration was done, what kind of template was applied, and in addition would keep the information about the sequence, so it still has the information about multi-band factor of 8, or like this is the like run 1. Fmri prep is not the last step in the preprocessing, because such things as high-pass filtering or regressing out so-called like dummy TRs could be done after Fmri prep is finished. So we did exactly that, and the output we have chosen not to have in the bits format. So I just want to stress out that let's say having things in the bits format is convenient, but it's not really required to get reliable results. So we have chosen not to use bits format in this case because the file name would be like huge, too long. So we kind of used slightly simpler naming convention. And now we get to a statistical analysis step. Like when preprocessing is over, you can actually get to the analysis that would answer your hypothesis. So for this study, like first big part of the statistical analysis is to like analyzing or giving at least some understanding of your sample. So in our sample, we collected healthy controls, meaning that those individuals would not have a family history of mood disorders, and they would not have individual history of mood disorder. So UD is unipolar depression group, and I call it, there's no diagnosis unipolar depression. There is the diagnosis of major depressive disorder or persistent depressive disorder, but because we collected both diagnoses in the same group, I would call it unipolar depression, which suggests that these individuals have never experienced mania episodes. So what's shown here are subjects that passed all quality assurance based on the behavioral, motion, and quality, image quality. 48 healthy controls, 42 individuals with unipolar depression. Gender female prevail. That corresponds to kind of like epidemiological statistics of the number of female versus male that suffer from depressive disorders. Among unipolar depression diagnosis, we had 26 individuals with major depressive disorder. So major depressive disorder is characterized by the episodes of depression. For example, for 2 months, the person would be depressed, and then they would feel much better. They will be euthymic sometimes for months, sometimes for years. Persistent depressive disorder is kind of like a milder condition, but it often lasts for longer than 2 years and does not have this spikes when the person is literally not functional, but still very debilitating in terms of cognition and functioning in general. Okay, so participants were around 30 years old, 28 plus minus 1 year. Samples did not differ from each other in terms of age. BMI was slightly above 25, so I just want to remind that if BMI is less than 25, that means normal BMI. Between 25 and 30, it's called overweight, and over 30, like 30 and over, it will be obesity. So our subjects were on average slightly overweight, just slightly above this magic number 25, which I think also corresponds with kind of population numbers. among the people who were overweight and obese, we had like majority of overweight and slightly like smaller number of people with BMI over 30. And one reason for that, it's difficult to scan people was the high BMI. Like first, sometimes they don't really fit well on a table. And second, when you need to close the like radio frequency coil, a person with a wider face or with like short neck often feels very uncomfortable, and in some cases you cannot even like close the coil. So functional magnetic neuroimaging unfortunately is limited in terms of how obese one can be to be scanned. Patients were slightly smarter than healthy controls. Their IQ based on NARD was slightly higher, which was significant. And VAS is the questionnaire that we administered before the scan that would ask our participants about how hungry they are, like six questions: how hungry you are, how much you would like to eat right now, how full you are. So based on those questions, healthy controls and individuals with depression were really at the same level of hunger, so none of them would be considered hungrier than the other one. So on the right, there is the distribution of depression severity against BMI. And as you can see, this top density graph shows that, of course, healthy controls were significantly less depressed lifetime than individuals with unipolar depression, and there was a pretty wide spread of scores in depressed individuals. So what I plot here is what we call spectrum measures of depression. They measure it by the Moods SR questionnaire. This is a long self-report over 160 questions where people answer about, like, have you experienced this particular symptom or this particular situation for the period of longer than 3 days, like ever in your life? And in this way, they describe the experiences with depressive symptoms. It's called spectrum because, in fact, it's not kind of like two poles here, like depressed or not depressed. Individuals with depression like really differ in their experiences with depression severity during their lifetime. And some people would have these experiences often, but they would not be very severe. Other individuals would have much more severe episodes of depression. So to measure the burden of depression across like all individuals, we use this RDoC approach when we look, instead of looking like between the groups, we look across the diagnosis. So looking across the diagnosis, we can tell that in healthy controls, which are in blue here, there was no any relationship between depression severity, and even healthy people sometimes have sub-threshold symptoms of depression, and this is what we measure it here. But anyway, there is no relationship between BMI and depression severity. And in patients which are in orange here, there was a significant correlation, I would say partial correlation, after we accounted for sex, age, and IQ in these individuals. Now, the second very important part of statistical analysis is the subject level analysis. So we use this Fmri prep and post Fmri preprocessed files as inputs. We used FSL to build our first level models. For explanatory variables we used in each subject model. It's food anticipation, object anticipation, food processing or food rating when they say pleasant, unpleasant, and object processing or object rating when, again, like they say if object is pleasant or unpleasant. Contrast of interest, because we were interested in, so like looking just at food would be kind of unfair, because let's say some people might activate their brain more during any kind of anticipation, other people less. Therefore, we used object anticipation and object rating as the baseline and compared food against that baseline. So the contrast of interest was food versus object anticipation and food versus object rating. So two two contrasts. So at the group level analysis, we used sandwich estimate or SWE tool in FSL, which we applied in the inclusive mask, which I mentioned later. And two types of corrections were used. Like first is family-wise error correction that we were able to apply using 5,000 permutations and TFC. So that threshold was set up at 0.05. And then because we had four contrasts of interest, so we had food versus object anticipation and food versus object rating, and we kind of modeled it as like +1 and -1, suggesting a different way how depression and BMI could interact. So we had to adjust for four contrasts, which gave us corrected p-value of 0.0125. So inclusive mask included all the voxels that responded by increasing in activation to either food anticipation or object anticipation or food rating or object rating. So like any voxel that responded in one of these conditions would be included in the mask. And again, like we used our dog approach that helps to account for within group variability. So across all BMI values, we did not split subjects into normal weight or overweight or obese. And also across spectrum lifetime depression. So the model would look something like that brain activation during food versus object anticipation would be explained by the interaction between BMI and depression score, lifetime depression scores, and one accounting for age, IQ, and sex. So in two models. Now this orange mask over here shows this inclusive mask that we used. So like indeed, like many voxels in the brain were actually involved in anticipation or like rating. Not all of them though. In this mask, there was one region. This is a right inferior frontal gyrus that was significant, that responded to BMI by depression interaction. And this 3D graph on the right shows that the patterns for people who were depressed and heavy or lightweight but depressed, like significantly different from those observed in people who were overweight and obese, and at the same time I had experience with very severe depression symptoms lifetime. So further, to kind of simplify the presentation, I plot here the interaction as a function of group. And what we see here, so like this is the right inferior frontal gyrus activation, the same as that purple blob that I showed in the previous image. So this value here is the difference in activation in this region between food anticipation and food and object anticipation. So positive number means that people activated the brain more when they anticipated food versus objects, and negative means that people activate the brain less for food than for object anticipation. So healthy controls, which is on blue, it did not really depend much on BMI. And in patients, we can see this significant trend that individuals who were lightweight, so like under 25 units in BMI, would have positive relationship, suggesting they activated the brain more when they anticipated food than objects. And heavier individuals with depressive disorders would do the opposite. They would deactivate brain for food and activate for objects. So then when we extracted percent signal changes from this brain region, We also decided to check how it correlates. We did not find any significant results for ratings, so, like, by the way, so that was the only significant result, like food anticipation versus object anticipation. But then when we did the statistics on the extracted percent signal changes, we found a significant negative correlation in both group and you know, like when you look in one group across all the subjects, suggesting that higher anticipatory difference between foods and objects would result in a lower difference between objects and food during making pleasant and unpleasant judgments. And just to remind, like this piece here is for heavy patients, like obese and overweight, or like over 25. They have low, well, I would say like negative anticipatory activation, and those people would have a positive difference between objects and foods, suggesting that they activate more for food than for objects when they need to make pleasant and unpleasant judgments. So in the next analysis, we try to see how this brain activation, or these differences between processing of objects and food would be related to ratings of how much they like items and how much they want food items. And I need to tell that people in general liked food more than they wanted them right after the scan. So that's why the scores here are lower for want ratings than for like ratings. And I would like to remind that 9 would mean like, I really like it, I really want it, and 1 would mean I don't like it and I don't want it. And we found that the group moderated the relationship between the difference in food versus object activation in the right inferior frontal gyrus and like and want judgments that people made about the food items right after the scan session. And interestingly, in patients which are in orange, the higher difference between foods and objects one would have, the lower ratings of like and want they would report. So based on this, we came up with the empirical model where BMI and depression might be related. And this relationship, this interaction would affect food anticipation in the right inferior frontal gyrus, which in turn, and I think this is a causal here because anticipation always happens before actual processing of food-related information, in our case, pleasant, unpleasant ratings. So food anticipation would be related to processing of food-related information in the same brain region. And then how much people like and how much people want food would be also related to activation in the right inferior frontal gyrus during processing of food items. Here we cannot really talk about the specific directions. It's always possible that people like that food, that's why they activate more or less for that specific item. And that's why we find this relationship. Or in contrast, people make the judgments, they activate the right inferior frontal gyrus, and then when they make the judgment, this determines the score that they give to that specific item. So this relationship can be kind of like bidirectional. So to give a little bit more, I guess, like to summarize how this model was supported by the results. So this is the first figure that I showed, which suggests a partial correlation between depression severity, lifetime, and BMI in individuals with depressive disorders. Over here, BMI and depression interaction would be related to magnitude of activation in the right inferior frontal gyrus for food versus objects, and objects we used as a baseline here. So when BMI increases, then anticipatory processing in the right IFG decrease. And in the red square over here, I show heavy participants. So I'll keep this, like in the model, like in general. So these like heavy individuals who also happen to be patients. Now, like these individuals in the red square, they have low or decreased activation in the right IFG during anticipation, but have increased activation for food items during processing. And then when they need to make judgments about how much they like or how much they want, like surprisingly lower ratings compared to the like less heavy counterparts parts or like healthy controls. I mean, like it's known that right inferior frontal gyrus is responsible for, like formation of like emotional impressions and also for control and for conflict resolution. So it is possible that this is the mechanism that depressed individuals use to trying to control the appetitive behavior. So they try to decrease in activation when they see the food item in the right inferior frontal gyrus to kind of make this judgment that they don't like or don't want this food. But given the amount of stimulation, like food-related stimulation that we have, if one constantly inhibits processing or like normal response to food items, it might become very stressful and it might induce reactions that lead to a depressive episode. And when it did in the depressive episode, it's really easy to gain weight because it's not only mood, it's also dysregulation in sleep and appetite in food consumption. So the study conclusions are that interplay between depression and obesity and unipolar depression is related to activation pattern in the right inferior frontal gyrus during food versus object anticipation. That these differences between food and objects during anticipation are negatively related to the differences between food and objects during rating of actual food and object items as pleasant or unpleasant. Depressed individuals who increase the right inferior frontal gyrus activation during food rating had lower ratings, like and want scores, than participants who decreased in right inferior frontal gyrus activation, which suggests that they might try to inhibit or control bias toward food and increase also like their emotional response to it. And we think that our results might suggest that this activation pattern for food versus object use may suggest a risk factor for both weight gain and worsening of depression in individuals with depression disorders. And we think that it is possible that this might be a novel target for intervention if somebody is at risk to be obese, being already like diagnosed with depressive disorders. There is a possibility that you can change you can change response to potential food items, response when you don't have food available or you don't have even food cues available. So when you encounter the cue, your response would be more optimal. So now, like the last step in the ReproNim process is publication. And publication includes not only publishing the paper, but also data sharing. So like one way to share data, this is what we actually use, is to submit all the data we collect to NDA. And it became easier using this UTConf and ReproIn heuristics together with bits to NDA script. There are like downsides to this script. It's not always works as intended, but you know, like with certain changes, you can make the submission of neuroimaging data pretty easy using these tools. And like again, like this work was just completed. We just finished writing the paper. It is not yet published, but we submitted a preprint, which is kind of like our first attempt to summarize the results. Like, of course, we continue like working on this, so we might get a second version. But I guess what's important that these versions stay in the meta-archives. So whenever like a researcher would like to look at our progress with this study, they able to look at different versions and see like if we got more subjects or if we decide on a different data analysis. So like this part is documented. So now to summarize ReproNim advantages, I can tell that UDConf and ReproNim taken together makes much easier initial data conversion and makes much easier deposition to NDE archives. Containers help use consistent versions of software that could be different for different studies on the same computer. And I'll give example. For example, if I install FSL system-wide, I will be able to use that version for like all my studies. But in case if I like to use different versions for different study, then I would need to you know, do like some manipulations. So in some cases, it would be even like impossible. Using containers inside each study folder makes it relatively simple to use one type of software or like pre-processing pipeline for one study and a different type of software or different versions of software for different studies. Containers can also be very successfully used in the cluster environment, especially in the cluster environment with limited or no qualified support for neuroimaging. So not all universities have a good support for, you know, like neuroimaging work like I do. Very frequently, people who support clusters don't know much about software that is available, and sometimes they just refuse to install specific software because it can alter dependencies. So like inside the container, inside the, let's say, Singularity container, you can really define your environment and be basically independent from the environment set up on a cluster. There are, of course, specific needs. So we kind of like talked to Eric and David about that. And like one big need is study node and management system that would be directly linked to the study folder or this participant's TSV file. Why this is important is that, I mean, like ideal scanning environment does not exist. Sometimes scanners crash and you would have 2 scanning sessions instead of 1. This should be documented and all the pre-processing pipelines should account for this. Let's say this interruption in the scanning session can result in a, let's say, larger time frames in terms of, let's say, if you're doing coding in memory test and you plan to have 12 minutes between coding the test, well, sometimes you might have like 40 minutes because the scanner could not reboot. It would be nice to have this as a more like automated metadata and annotation system. So when I start analyzing the data, I would have a list of flags, or I don't know, like the problems with scanner or the problems with specific subjects would be highlighted and popped up, or, you know, like right away when you start the data analysis. And it would be nice if implementation of data-led would be smooth or smoother, at least for people who are familiar with other programming environments, such as Shell, for example. Because it seems that even though data-led creates new opportunities to not only to manage the data, but also track the changes in the data, sometimes it's time-consuming to learn how to actually use it. And I would like to say thank you to people who were involved in this lab, my lab, and my faculty collaborators, and also to NIH for funding this research, and also for funding ReproNim research. And I guess at this point we are ready to take questions. Thank you.
That's great. Thank you very much. Does Jarek have any commentary he'd like to throw in there as we prepare to take more questions from the audience?
I think I'll just maybe first resort to the audience.
Okay. So the audience is invited to carefully unmute themselves and ask questions if they have.
Hello.
Excellent.
Can I hop in?
Yes, please do.
Hi. Yeah, just here for my first ReproNim seminar. Let me also turn on my camera, a little personal. Yeah, so here for my first ReproNim seminar. Thank you for the interesting discussion on obesity and reproducible workflows, an example of reproducible workflows. And my question for you is thinking about simultaneously the aim of reproducibility in science to accelerate innovation, make it easier to converge on that local minimum of whatever truth that is for a particular question or program of study. And the other aim of also actually contributing to treatment, contributing to real world applications, right? And I think that depression and obesity is a really interesting use case because if we think about how it's treated, it's an individual one-on-one treatment process that occurs with the care provider and the patient. And there's some program that the patient goes through to, in determination of what the prescription of the treatment should be, whether it's medication or whether it's exercise or some other recommendations. Now, your results are captivating because they suggest that, okay, well, perhaps there's a place in the brain that might change adaptively to environmental stimuli. And if a clinician had access to this patient's information, maybe they'd be able to make better informed decisions regarding their treatment. So my question is a very long-winded way of saying, how do you envision the current reproducible workflow empowering and making something like Francis Collins, the old director of NIH, I don't know if he still is, I know he was acting director a while ago, make his vision of a personalized medicine initiative actually possible? So how can we go from having data that we're depositing into kind of this this gated community and NDA, making it accessible to clinicians and patients that want to leverage their data for actual use in the real world?
Yeah, this is a very important question, and I don't think at this point we are ready to go to right to clinician's office, and maybe not with functional, MRI. I do envision it's possible with near-infrared spectroscopy, when you can actually have a person in office, and this methodology could help track activation for a specific stimuli. Let's say if you constantly suppress processing of, like food items, or like if you constantly activate your, like in your frontal gyrus, trying to resolve this conflict when you want this food item, you want this piece of steak, but you know that you should not want it. Like how you moderate it? Well, maybe overexposure would help it, or maybe biofeedback will help it. I mean, like neuroimaging is great, fMRI. I mean, it can pinpoint the region, but I don't think it can be actually used in the office, because we, like this is a snapshot, right? Like this is the person, like right now, right here, they come to doctor's office very frequently at a different environment. And what I found in clinical science that even when you do clinical assessments, it's very variable. Some people really want to help, so they're looking at the clinician for little science, like what should I respond to make you happy? Other people, in contrast, refuse having symptoms. And we do have, like in a study, like one that's a longitudinal study, let's say we can have a person who comes for the first appointment as a healthy control, and at the end of the study, they would be diagnosed with major depressive disorder, well, just because they never disclose their symptoms. So people are very different in how they disclose symptoms. So that's why some portable way of measuring brain activity would be much more appropriate for in-office interventions. Like, of course, when we know a little bit more about like what happens, like if you were depressed, well, maybe not currently, but if you have experience with depression symptoms, were diagnosed with major depressive disorder, for example, like what do you need to do not to become obese? I mean, like depression by itself is a very debilitating disorder. I mean, like obesity by itself is not good either. You don't have both at the same time. So, I mean, like maybe without making like global, aim to treat everybody from everything, maybe just help people not to gain weight during the depressive episode. Or in contrast, some people with, obesity, then they have a better chance to become depressed eventually. So like, what should we do to maintain the level of functioning, to maintain the level of like normality, like normal mood? So that's the important question. But like as a researcher, I think that even if we could standardize the outcomes in some way that could be readable by other clinicians, that would be great already. Because what happens when you so like if you do like a large assessment, like let's say SCID, the clinician would be sitting with like a fat, like book with the questions. They would be writing, they would be like circling, and then not necessarily like in 10 years, it would be clear what was there about the patient and this. So like this are notes for a clinician to make a diagnosis. So like for me, like standardizing the outputs, they're structured interviews, so like they're standardized to a certain extent, but standardizing the output, so any person who would look at the notes, and that's why we talk about like metadata, that's why we talk about kind of like getting this in more standardized format that would be available in the maybe participant even folder. That would help. Like we know we did this assessment, somebody else did the same assessment. What we don't know if it was done in the same way. Like, so that's a big problem then for the data analysis, and that's this is something that could influence reproducibility, not because you cannot reproduce the effect, but because you don't really know what to reproduce. So if I'm clear what I mean.
I think that ties into this generalization versus reproduce in this data, or how well does that finding generalize across the different types of important characteristics of the subject population, I presume.
And if I could chime in, if we are talking about metadata, quite often we don't know what auxiliary data might come useful later on. Let's say when this questionnaire is asked, right? What was the weather, right? What was the temperature in the room? All those factors might affect the results, right? And contribute to the variance, which we cannot explain, but this data is not gathered. So with automation of data gathering, which would contribute also to reproducibility by reducing the variance, but also it might just provide, new means to figure out new effects which we didn't even think about. And then they would be used in clinical settings because, oh, apparently, indeed, day of the day really affects, how humans answer, right? And maybe we should account in which mood they were when they entered while answering that questionnaire.
Other questions from the audience? While we wait, I may throw one in. So you talked about using the NDA. Was that principally for sharing the sort of not the raw data, but the acquired imaging data? Or are you also thinking about, sharing the derived, processed results themselves? And would that be through NDA, or is there other ways that you consider sharing the derived imaging-based results?
Yeah, I think it's a pretty time-consuming in NDA right now. So I think they have mechanisms to submit to clinical data with kind of like preset templates and stuff. They have this specific script that, I mean, like we had to do some work on that to make it work. So this is specifically for raw data in the bits format. But I don't think they have anything clear and specific at this time for derived data. At least I don't know about that. But we do consider after this paper is published to submit this data either to OpenNeuro or maybe like OSF or some other place, because in fact I think it is important to have this like pre-processed data you know, available so people could actually replicate your findings.
Yeah, I think that's a good point. And part of the reason of the question is that when people do go to replicate one's finding, it's also good to know, to have the original results, to know how well you've succeeded at, you know, starting from where you ended, so that when they move on with additional data or additional approaches, they know that they're starting from the same place. And so an OpenNeuro, of course, comes to mind, which was sort of a leading question to OpenNeuro. Is a great resource. OpenNeuro also does permit us, or permit one to share NIDM representations with those derived results. Now, I'm going to guess, and others may fill in, that, you know, our support for NIDM representations for any sort of random, you know, functional experiment may still be in progress. So I don't know how, if you've run into any of the, even tried, you know, to create the NIDM representations that follow along with your statistical analysis of the Fmri data or not. And it may be premature to even ask that, but I figured I'd try.
Yeah, well, I think that you can always supply the scripts you use. The most important thing here that, I mean, like, we need to script what we do. And when you have those scripts, even if there's no platform to post those scripts or present them in a consistent manner, you still can post them on OSF or somewhere else, and it also contributes to, you know, like reproducibility. Even though it's not, I mean, like, I find this like BS format is very convenient, very well described, and people can, you know, use like old data and then rename them and create a bits data set, or even like reconvert. And this is what we did, like, with kind of like legacy data that, you know, like includes all like 500 subjects and I don't know, like 2000 scans and so on. Like we like literally renamed, reconverted, and pre-processed them. So this data would be available in the comprehensible format right now, like, and people would not spend 2 years trying to figure out if this is a first session or second session, or what kind of file is that, or does it belong to, you know, this group of subjects or that, or if like even like parameters, it's useful to know if that was like a regular acquisition or multi-band acquisition. So I think it's all like, even if it's not available right now, if it's stored in the appropriate format and if it's done in a kind of like scripting fashion, one can then convert those scripts in a more consistent framework when it's available.
And again, I think that's a great point to emphasize, that even if there isn't an exact standard for everything, the more, you know, precise we can be about making all the elements available somehow, even though that's better than, you know, not doing that at all. And so again, we don't necessarily have to get the whole way in every project, but the more we can move a little bit, you know, per project as the technology evolves, that's a great advance. So I see we're pretty close to the edge of our time. Any other burning questions out there?
I just want to emphasize on the last phrase you've made as my commentary. As you could see through Anna's presentation, she did not necessarily use all the great technologies we had, because there were some maybe inconveniences, right, and learning curve, but indeed learning some, right, not maybe all of it at once, right? You could go piecemeal, right? You could learn one trick and use it, and that will contribute to make your study better, right? And then you in the next one, you go to the next one, right? And just gradually increasing your reproducibility on this tag.
Yes, that's a great example. And again, many of our projects are partly underway anyway, and so you can't do everything when you start on your flying plane. Okay, so thank you very much. I thank Anna and Jarek for this great example. And again, it's a good example of some things able to be used, and also a pointer to the things that really still need improvement from the ReproNim point of view, and more accessibility to being used. So that's kind of what I was trying to get at, sort of adding the pros and the cons, in this kind of format. So again, thank you for perfectly illustrating some of the examples that I think is good for the community and for us to see.